Hello Biology 300 students, Mr. Parker here, and in this screencast we'll be breaking into two different segments actually. The first segment that I'll be doing is on the genetic material, the requirements for genetic material, and then also some of the scientific experiments that proved that DNA was the actual molecule that contained the genetic information. And then the second segment that you should watch to help fill out your notes will be the segment on DNA structure and its function. And you should have out your notes. The packet that I've given you with the yellow cover sheet will help you do the information for this. And also you should have your packet that I passed out in class. We'll be referring to a few diagrams in there, writing a few notes on there that I would definitely have out with you and handy to do. So make sure you have your packet with you and your notes, okay? So moving through this screencast, as you can see, we're going to cover basically the first three bullets. The first segment is going to be on the requirements of genetic material, and then also DNA as basically the discovery of DNA as the hereditary information, containing the hereditary information, and then we'll be talking a little bit about DNA structure. And then in a later screencast next week, we'll go ahead and talk about DNA replication. Okay, so this is about time you want to have your notes ready to go, kind of get moving. We see here, to be considered genetic material, there's three requirements. They must be able to encode for the production of traits. Okay, so key here is when we're looking at must be able to encode for production of traits. And you might say, well, what does encode mean? Okay, basically it needs to give instructions, okay, for the production of traits. And you have like traits or characteristics that are passed along from your parent, from parent to offspring. The second thing here, it must be able to replicate itself. Okay, and if you think about DNA, we're talking about replication in order to be passed along to new cells through the process of mitosis, and also you need it for meiosis. And then the third thing they must be able to do is control. They must be able to change in a controlled, change in a controlled way. All right, meaning the cell has to be able to control its growth, because if it doesn't control its own growth, it's considered to be what we call cancer. Okay, so these are the three requirements that your genetic, in order to be considered genetic material, it has to have these three different requirements. It has to be encoded for production traits, be able to replicate itself, and also change in some type of controlled manner. All right, so moving on from here, and kind of giving you some a little bit of brief history about DNA. DNA was discovered, the actual molecule was discovered in 1868. Now remember, DNA stands for deoxyribonucleic acid. All right, so back in the late or mid 1800s, DNA was discovered. Now you need to remember it was discovered, but it didn't. The scientists at that time didn't know the importance of it yet. They knew it was a molecule within your body, but they didn't know that it was going to carry all the genetic information that we know of now. All right, and that actually will be, like 80 years later that they will actually discover that DNA contained the genetic information. There is a book, a piece of work that was out there in 1915 that came along called The Chromosome Theory of Heredity, and that was published in 1915. Okay, and this was by a scientist named T.H. Morgan, and he did a lot of his work on fruit fly heredity. Okay, and what Morgan proved through this, through his publishing his work and his scientific experiment, was that he showed that chromosomes, behaved in accordance with Mendel's law of genetics. And Mendel is what we say is the father of genetics. He did all his experiments, and when we get into genetics, we kind of see the importance of what he did. But chromosomes and how they're passed along from parent to offspring, and the numbers and all that stuff, all work out due to the fact that it follows the laws of Mendel. So with T.H. Morgan's work, he proved that chromosomes, had the material in it that carried the genetic information. Now, chromosomes are made up of a couple of different other substances called DNA and protein. So now the scientists knew that chromosomes were containing the information, but out of these two substances that you see, or molecules, you have DNA and protein, they were not sure which one actually carried the genetic information. So at a time, in the early times, these scientists, so you're talking between 1860 up to like this time in 1915, and all the way up to about, through the different experiments that we'll look at, they believed, scientists believed that proteins actually carry the genetic information. And really the only scientific reasoning behind it, that your body or the chromosomes were made up of 60% proteins, and your DNA was only made up of 40%, or your chromosomes only have 40% of DNA. Okay, and you can see here there's 20 subunits that make up these proteins. These subunits are called amino acids, and DNA is only made up of 4, which we know are like your nitrogen bases, your A's, your T's, your G's, and your C's. Okay, so because scientists are seeing the percentage difference and also the number of subunits, and they're thinking the importance of this DNA structure, of the genetic information, they assumed, you know what, it's got to be protein because there's more of them, there's more subunits, and there must be, you know, for all the information that it codes for, it has to have all these subunits in order to get the right information. Okay, so then, you know, you're talking 1915 with the chromosome production, T.H. Morgan put out there. Then there's another scientist that came about in 1928. His name was Frederic Griffith. Okay, and kind of getting some of this basic information up here for you. You can see what Griffith did. He said some factors causes avirulent bacteria to become virulent bacteria. Basically, in his experiment, he was looking at these viruses, okay, that were, bacteria that were, some were killing organisms and some were not. And then what he found was organisms that were at one time didn't kill could now actually kill an organism. And what he came up with in this idea is called transformation. And basically he was saying that DNA could be transformed from one to the next. And he discovered this mechanism, okay, through his experiments that you can see that he did here with the mouse. And you can see that here in this diagram that we have here, okay, and you can see the different setups that he has. I'm actually going to switch over to a page in your notes on page 35 in your packet. So if you can flip out to page 35, I'm going to exit out of here real quick and pull up that. Okay, so as you can see here, okay, what we're going to look at is the mouse. Okay, here is looking at what the scientist has done is he takes this type R, okay, which is a non-virulent, so it's not a harmful bacteria, and you can see here what happened is that the bacteria was still living. Okay, then he treated it with an S virulent bacteria, virulent being deadly, and what he noticed, obviously, you notice that the mouse dies. So what Griffith decided to do here was he was going to treat this S-type bacteria that was deadly with some heat so it couldn't reproduce. All right, he injected it in the mouse, and what he found is that there was no bacteria and the mouse was able to live. So now this deadly bacteria at one time was no longer harmful. So then he decided, you know what, here's what we're going to do. We're going to mix up, we're going to take that type R, which again was non-virulent, okay, it wasn't harmful. He took the heat-killed bacteria, which wasn't harmful at the time, okay, because we saw that through this third step here. And what he noticed was by combining these, injecting in the mouse, he found that the S bacteria was able to be recovered, okay, and was actually be able to transfer its genetic information from the S bacteria into the R bacteria, and now that R bacteria was producing deadly bacteria and killing the mouse. So for Griffith, this proved the idea that transformation of the genetic information was occurring from one organism to the next. Okay. So Griffith, 1928, came up with the idea of transformation. Then there's another group of scientists that came along. Okay, Avery, McCarty, and McLeod. You know, a number of years later, 15, 16 years later, and what they looked at. Okay, they used some enzymes to break down the cellular components of transformed cells. And this enzyme is called DNase, and stopped. What it did was it was stopping transformation. All right, and what he had done, or what they have done, is that they took this S strain of bacteria, okay, and they put it into some of the macromolecules that we had talked about earlier this week. Okay, lipids, carbs, proteins, DNA, and RNA. Okay, the different types of nucleic acids. And what they had found was the only thing that transferred from one to the next was that S bacteria, okay, through the DNA structure. So the conclusion, as you can see in a nice yellow box for you, that the molecule that carries the hereditary information was definitely the DNA. So Avery, McCarty, and McLeod was basically reinforced the idea of what Griffith talked about in the idea of transformation, but they also proved the idea that DNA is actually the transferring information. Okay, another group of scientists, Hershey and Chase, a number of years later, came about, and they were working with something called bacteriophage, and these were actually viruses that were attacking bacteria. Okay, as you can see here, the active component of the bacteriophage that transmits the infective characteristic is DNA. So through their experiments, okay, and their lab, they came up with the basic conclusion here that DNA, again, is the transferring factor. So they basically reinforced the idea of what Avery McCarty and McLeod talked about. So if you look at their experiment, okay, here in this particular one, what they were doing is they were tracing or labeling protein with sulfur, and on this one they were labeling the DNA, as you can see here inside, with phosphorus. And what they did was they put them together. You can see here how the virus is attacking the bacteria. They blend them together, and what you notice at the end, okay, in here, is that your DNA actually carries the genetic information from the virus into the bacteria. So through Hershey and Chase's experiment again, they're proving the idea that DNA and genetic information are correlated to each other, and DNA was carrying that genetic information from one organism to the next. Okay, so these were very important experiments that proved the idea that DNA was in fact the genetic transferring information factor, whereas DNA had all the requirements. Okay, it was able to control its own growth. Okay, it was able to replicate itself, and it was able to encode for its own production. So that fit the requirements of the factor of being genetic material. And also through these three different experiments by Hershey and Chase, Avery, McCarty, and McLeod, and Frederic Griffith, the idea of DNA transforming the genetic information from one organism to the next. Okay, so this was Screencast 1, the segment 1 of the Essential Dogma. The next one that you'll be seeing is on the DNA structure and its function, and that will give you a better idea of basically what the structure of DNA is all made up of, and what builds it up, and how it's a double helix, and all the information that goes along with that. So this is Screencast number 1 for Essential Dogma of DNA and the genetic information and genetic material.